for any other speaker, I would now be making loads of jokes about the next speaker. But since he's my boss, I can't actually do this. So I'm afraid it's going to be a boring, bland introduction. Janet, it's my pleasure to introduce you. She's the director of the European Bioinformatics Institute, and she had the wisdom to recruit me a couple of years ago. Whoa, I haven't finished. But not that boring. So while I won't go into her CV, I did realize when I looked it up that actually Helen and Janet have quite a few things in common. So they both started out studying nucleic acid structure. They both have long and distinguished careers in structural biology and structural bioinformatics. They both have a brand new building. Yeah. And they both are part of a very secretive Freemason-like organization, which they themselves call the Old Girls Network, and which I always thought was like 2 or 3 of them. But Janet sent me a list of names, and it's spread out everywhere. It's a very big and secretive organization that I'm sure they don't want me to talk about. But it's my pleasure to introduce Janet, and she'll talk about. Yes, you're now allowed to come.
So it's a great pleasure to be invited here to celebrate with Helen. It's wonderful. And let me work out how to work these implements. Hang on. I think that's. There we go. So what I thought I'd do, which is rather different, over the course of our careers, Helen and I have sort of collided at various stages in various different ways. And so I thought I'd sort of go through it, in part because it reflects Helen's contributions, but also because it sort of reflects the way that the science has changed as we've gone through. As you've heard, Helen very much started off with the nucleic acids. And she always tells me that this, it's, your first love is always your strongest love in a way. And I think that's certainly true of Helen. And I started, as was already said, by looking at dinucleotides. And we've heard very beautifully from Steve how Helen was one of the first people involved in crystallizing. Not, in this case, I was looking at NAD, but Helen was looking at APA or UPC, UPA, whatever it was. And this was one of the first things. And so some of the first papers I looked at when I was trying to understand the conformation of this beast, both computationally and also experimentally, was some of the data that was coming out from crystallography on the nucleotides. But then, unlike Helen, I left, and perhaps my first love isn't my best love, because actually I find proteins just so beautiful. And I started to look at protein structures. And of course, at that stage in the 70s, this was when all the new structures were being determined. In terms of DNA, there was really quite only a few. There were hardly any structures coming out. But for proteins, the number of structures being determined. So I started my PhD roundabout here, and it was just at the cusp of when all of these new structures came out. And so the challenge that we faced was really to look at some of the basic principles of protein structures. And we did, along many other people as well, looked at different aspects of the motifs, the sorts of motifs that we found in protein structures. And initially, of course, we did it by all writing Fortran programs to try and abstract this information from the data. And gradually, this got more and more sophisticated, so that now you can find all of the beta turns in the whole of the PDB with a quick flick and look and find all of this information. And we also got involved at that time, and this is where we sort of move on, in terms of validating structures. Now, this was nothing to do with trying to find mistakes. It was purely to do with trying to understand the stereochemistry of the main chain and the side chains of proteins. But as part of this, and this really is the work of Roman Laskowski here, we developed this program, PDB Clean. And that was because at that stage, this was in the late mid-80s, we had to do a lot of work on cleaning up what was in the PDB. And I think, this is a typical, I think, Roman name for a program. It was very obvious what it did. And I think as a consequence, really, of the name of this program, I was asked to sit on the SAB for the PDB. And so we sort of started at that stage then I came over to Brookhaven and then began to get to know Helen. Now, after that, we actually began to work on nucleic acids together. And I think Helen could see what the sort of stuff that I'd done on proteins, and she knew that she very much wanted to do the same sorts of things with the nucleic acids, and in particular, to do with the NDB, the nucleic acid database. And in particular, looking at DNA binding proteins. That was really what she was interested in, and looking to see whether we could understand more together with her knowledge of the nucleotides and my knowledge of the structures, how these things might fit together. And I think this is the way that the whole field evolved. And so as part of this grant, we looked for various DNA binding proteins, and we went through all these different motifs that were then in the field and developed an idea of all the different ways that proteins can bind DNA. And we also put together a sort of a catalogue of the structural family of DNA binding proteins at the time, at the end of the 90s. And you can see here, and we heard from Steve about the way that different ways that small molecules can interact with proteins. This just describes some of the different ways, and you can see how different they are, that proteins can interact with DNA. And so we made a sort of a structural family of these, looking at them and trying to understand what happened. And this is just one. Obviously, the largest group remains the helix-turn-helix group. It's very common in transcription factors. It binds in the DNA major groove, and you can see it here. And there are various variants of the helix-turn-helix. In contrast, of course, we have the zipper proteins here, where we have the two helices that bind in sequential major grooves. And so you get a completely different sort of recognition between the protein and the DNA. And so as part of this, we wondered whether we could identify DNA binding proteins just from the structures. So the idea was you go through, you look at all the structures, you find out what all the motifs are. Can you then go back and use this knowledge to predict ab initio, if you have the structure of a protein, and this was before the days of structural genomics, I should say, could we predict whether we could see find DNA binding proteins or not? And really, we had two different ways of doing it. First of all, recognizing the DNA binding proteins from templates of those motifs, and secondly, from trying to use patches and look at the electrostatics. And so this shows, again, the way that one gradually evolves new, more sophisticated ways to recognize the functions of proteins. And these were some of the motifs. These are all helix-turn-helix motifs. And we used the electrostatics so we could try to predict where we had DNA binding hotspots. These were the known interfaces, and hopefully these would be superposed. And broadly, we ended up with a variety of different programs that allowed us to predict with some success. I would say not total success, Helen. I don't know if you agree with that. We never really cracked the whole thing. We also, at that stage, this was when, you can see, we had 32 protein RNA complexes, and we decided we would do a study of these few complexes. And this was, in fact, after all of this, we ended up from this grant having 3 papers that were to do with the way that proteins recognize DNA and later RNA. Now, again, these studies that we did then, with the use of all the tools that are now in the PDB, we could probably do, we could get an intern over summer to do the same thing and repeat it and get much better data and much more powerful prediction algorithms in the process. But I think it was sort of a slow process of realizing what was necessary. And this is the acknowledgements at the time. I think it's interesting. Helen really doesn't look any different now than she did then. I mean, the person who looks most different, some of you probably don't know Nick Luscombe. He was the youngest person. He's the one who's changed most. But anyway, that was happening actually sort of at the same time as the changes that came around in the PDB. And I'd really now like to move to the PDB and how that has evolved. Clearly, during the 90s, this is the 90s here and the growth of structures here. And you can see it's just at the point where the number of structures, in fact, the flood of structures hadn't really started at that point. But at that point, at the PDB, there was a real challenge. We had new tools, and PROCHECK was among them. There was 2 or 3 other very well-known and well-used programs for validation that became available. Those really clearly needed to be incorporated into the PDB process. We went on various visits to Brookhaven as part of the SAB, but also, I've just put this picture up, which is actually, I think, from one of the IUCR congresses, but you can see Helen and Shoshana Vodak and myself here. And one summer, we spent some time in one of the huts in Brookhaven, I don't know if you remember the Brookhaven park, but you're sort of in the middle of nowhere, and we were in the middle of this wooden hut. It was like playing Girl Guides, I have to say. But we had a great time doing that and looking at the PDB and trying to work out how the PDB might adapt to the new challenges that it faced, because it was clear that some changes were necessary. In parallel, this was the time when the structural genomics things first started to get going. And this was a list of some of the, this is a very old slide, some of the structural genomics projects that were emerging. And so in the early and late 90s, there were some really major challenges facing the PDB. The first was this, what then we thought of as a flood of data. We realize now it was just a trickle compared with what we've got now. There was also a backlog. There were 100 PDB files out of the 500 that we had that had been sent to PDB but weren't yet available, because everything really was done by hand. And so there was the approach to handling data had to change, and that's very difficult when you're used to doing things in one way. The other thing, of course, was the emergence of the internet around that time. And that changed the way that we had everything that we did with data, how we deposited data, how we abstracted data. Cynthia was talking about the big tapes. I used to get big tapes as well, as many of us did. Of course, also, the fact that crystallography, when I started, there were a few labs and everybody knew each other. Whereas now, the whole discipline had spread worldwide and people were depositing structures all over the place. Also, the rise of NMR as a method to determine structures ab initio. And that introduced a whole new lot of challenges for the PDB, how to handle NMR structures. And of course, there was the issue, as there always is with databases, of funding. the funding for the PDB was split, well, between different organizations in the US. The US funded everything, and it was very clear that there was also a rise in the desire to really do some of this elsewhere in the world as well. And so the powers that be had a request for proposals to run the PDB, and Helen decided, and this was part of our, when we were in this cabin, it was about that time, that actually she felt that she would like to apply to be part of the process to run the PDB. And I think 4 of us were part of the first proposal to run the PDB, which was not successful. And I have to say to everyone, if you want to look for anybody with guts, look at Helen. Because after what was a very painful process, she went in again and did it all over again. And the second time, she was successful in winning the bid to run the PDB. And that was a terrific amount. I mean, the amount of work that was necessary for that, I cannot tell you. And it really, I think, set the scene. And because she really, really wanted to do it. And I think that is part of what's so important and why she's done it so well. At the same time, the EBI, the European Bioinformatics Institute, was established in about 93 near Cambridge in the UK. And when it started, there were 2 databases. The ENA, it was then called EMBL Bank, the nucleotide sequence database, and also we collaborated with the SIB, the Swiss Institute of Bioinformatics, on what is now UniProt. So there were just 2 databases. And it was very clear that there were very strong call within Europe that there should be part of the PDB, should be in Europe, and that it should be jointly funded. So there would be joint responsibility, and there would also be funding from more than just US funds. And so very soon after, in fact, I didn't work at EBI at that stage, I was a consultant, and we wrote a proposal to the Wellcome Trust to fund a few people to set up what has become the PDB in Europe. And so this then changes really the face of the PDB. And I should say that throughout this, Helen, who was herself at that stage just coming into this, was very supportive. And so we now see another facet of Helen's character and contributions, because what you see with databases that are distributed worldwide, and here I just talk about PDB Europe, but obviously PDB Japan is equally important, and it becomes necessary to work with many different people. And you see here, this over the time that the PDB was established in Europe, there have been actually, there's another person here, Phil Neal, that we were talking about. There have been a whole selection of different people that Helen has worked with and teams that have worked with. So now we have this distributed infrastructure around the world that I think is a model for any database that you want to talk about, because each part is different, each part has its strengths and some weaknesses, but together they work things out on a daily basis. And I know this, they are on the phone, on Skype, doing things all the time, every day. And so the ability of the people involved to work together, and here we see Helen and Kim, and that was what turned out to be, I wouldn't say a marriage made in heaven, but after a rocky start, it worked very well. And so these things do come together. And again, I think it is this working together that really matters here. And of course, the next step to establish the worldwide PDB, again, came with strong support from all components, including Helen. And she believes very firmly in this as a distributed infrastructure. And so I think Helen's contribution to setting this up and establishing it is really very important. And so within the PDB here we see, I can't remember when that was taken, but it's one of the advisory board meetings sorry, I've done that wrong. This is an in-joke. That's what comes of doing it last quickly, yeah. So just working together, I think, and that is what Helen is really very, very good at, and is what has made this whole collaboration work so well. But let's just take a second to look into the future. We know, this is just because I have this data set, but this applies. We know there are now over 94,000 different entries in the PDB. These curves show how all of the databases at the EBI are doubling. When you saw at the beginning, we had 3 databases at PDB, at EBI. This now shows each one of the black names is a completely separate database at EBI. So it goes all the way from the genomes and the variation through expression, through proteins, and of course the macromolecular structures, the chemistry, the small molecules, and down to the systems and the pathways and the interactions. The MSD and now the EMSD, this begins to cover the different scales in biology and will change and become more important. But what's very clear is that this needs to work together with all of this other information that we have out there. There's a project that has been SIFs that has been done again as a collaboration where we integrate effectively the data between PDB and some of these other, with UniProt in particular, so that we know that when we talk about ASP52 in the PDB, it corresponds to ASP52 in UniProt, which is where all of the functional information is held. There are still challenges, and in fact, this is an old, something old from that Gerard sent me. There's growth. There's the size and complexity of entries, and we know about the new format that's coming out next year and the new deposition and DNA system. Hybrid low-resolution methods, from molecular to cellular biology. And of course, the user base is changing from our expert small community of the 70s and 80s to a vast community of non-experts. So the increasing need for validation and in particular integration cannot be stressed enough. And the move from a structural biology archive to a biomedical resource, where this stays not just in research, but also in the biomedical community and beyond, I think is going to be a challenge that the wwPDB is going to have to face. And of course, what we want is the ability for knowledge extraction. We want to capture knowledge learned from the structure, and I believe some of this should go into the PDB using agreed standards, vocabularies, and ontologies. Some of this we have. There's some simple things like experimental protocols, biological unit, that's more or less there now. The role of dynamic movement, not yet there. Relationship to other entries, there are other databases, but these need to be linked more closely together, I think. And in particular, the functional highlights. We heard just now from Cynthia about this incredibly complex interplay between the different sorts of ubiquitin and their molecules. How do we capture that in a form that somebody can easily abstract it and use it for their own work? I think this is a huge challenge, and one that I think the PDB will grow into, if you like. And there are many complex things that we also need to think about as we're going forward. So really, I think if I wanted to sum Helen up, these were the words that came to mind. First of all, dedicated, beyond the call of duty. by any measure. Loyal to the people that she works with, she will defend you and always be straight and honest. Energetic, we know we know Helen's incredible. Oops, sorry. Oh, what happened then? Incredible energy for doing things. Her attention to detail is legendary. But if anybody knows Helen, that is, and she has an incredible memory and a filing system to match, so she can find anything from any time back. She's a brilliant organizer, which you clearly need to be. She cares deeply about the data, and that is probably the thing that has been most important in making the PDB a success. She cares about her colleagues, and for me, she's been, over many years, a good friend and colleague. So congratulations on your achievements in the first 70 years, Helen.